Today we're going to learn about the structure of DNA. DNA is a polymer of nucleotides. So a nucleotide has three components. There is a phosphate group, which is a phosphorus with 4 oxygens around it. One of them actually has a double bond. Go ahead and put a double bond on there. Then there is a 5-carbon sugar. In DNA, the 5-carbon sugar is deoxyribose. So the 5-carbon sugar looks like this. And if you remember back to when we did biochemistry, we can actually number those carbons. You go from the oxygen and you go clockwise, and carbon 1, carbon 2, carbon 3, 4, and 5. So the phosphate group has attached to the 5-carbon sugar. Then over here, the third component of a nucleotide is a nitrogenous base. So right here, I am going to connect it. I am not going to draw out the full structure, but if you remember back when we did biochemistry, we talked about purines and pyrimidines. This, you'll notice, is a double ring base. The base has carbons and nitrogens in this ring. And this, because it is a purine, this could either be A, adenine, or it could be G, guanine. So these are the components of a nucleotide. If you look at an entire strand of DNA, you will see the sides of the strand and the center portion of the strand. People often refer to this as a ladder. The rungs of the ladder, well, first of all, the sides here, this part right here, here, and here, that would be the sugar, 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 and in between are the phosphate groups. Same thing on this side. Sugars here where it connects in, and phosphate groups in the middle. In the center region, the steps of the ladder, this is where the bases are. And you'll notice that the steps are always the same width. That's because it's always pairing up a purine with a pyrimidine. So a double with a single. On this side, the single might be here and the double here, but the width always works out the same. If you think about it for a minute, if you put 2 purines together, your ladder would go out, and if you put 2 pyrimidines together, your ladder would go in. So the bonding is very specific. And you will remember, I'm sure, from Regents Bio, A, if this was adenine, this purine, this would have to be T. We call that complementary base pairing. If this was a G, G pairs up with C. So A and G are our purines, C and T are our pyrimidines, and this is complementary base pairing. The connection that they have right here is a hydrogen bond. There are usually 2 or 3 hydrogen bonds connecting the purine to the pyrimidine. Now, something else that is really unique, need a new screen, is that one side is anti-parallel to the other side. What that means is one side looks right side up and the other side looks upside down. So if I draw P for my phosphate group, here's my sugar, my 5-carbon sugar is here, and then 4, and then 3, 2, and 1. 1 carbon sugar being here, 3 carbon sugar being here. This side is, again, connected to a phosphate group, and then another 5-carbon sugar, 4, 3, carbon 2, carbon 1. And it keeps going like that. This side is numbered according to these carbons. So because the 5 is higher than the 3, we call this the 5' to 3' strand. So these numbers, 5 and 3, refer to this carbon and where they are in the molecule. Here will be my base. So I know it looks really tiny. In reality, if I had to draw all the molecules out, it would be much larger. But I'll say that this is my A, and here it is hydrogen bonded to a T. The T would actually look upside down, and the T would be connected it always connects into the one carbon. So here is my one carbon. But on this side, the oxygen is facing down. This would be my carbon 2, my carbon 3, my carbon 4, and the branch carbon 5. So you'll notice this is what we call anti-parallel, upside down to each other. The 3 carbon is connected to the phosphate group. The 5 carbon is connected to a phosphate group. This would be connected to the next one. So this would be my carbon 3, my carbon 2, carbon 1, connected to my oxygen, my carbon 4, gotta extend this a little bit, my carbon 5. So here would be the line separating one nucleotide from another. So here's one nucleotide, phosphate group, sugar, base. Here's the dotted line showing the separation, phosphate group, sugar, base. And it appears to be upside down compared to the one next to it. This one, phosphate group, sugar, base. So here's my base pairing with the hydrogen bonding, complementary. This side would be numbered 3 to 5 because the 3 is above the 5 on this side. So we call this our 3' to 5' side. This is going to be really important as we go through DNA replication and transcription and translation because the way that it's copied is very specific and enzymes are really specific in where they can work on a carbon. So where they can copy and make a new strand, we'll definitely be talking about 3 to 5 and 5 to 3.